Hi friends, in this video, we'll explore food microbiology, Module 1, the historical development of microbiology and microscopy. We'll take a journey through the fascinating history of microbiology, from the classic debate of biogenesis versus abiogenesis, to the germ theory of fermentation, and finally to Koch's postulates, which laid the foundation for identifying disease-causing microbes.
Long ago, people believed in the abiogenesis theory, also called spontaneous generation, which said that living things could arise from non-living matter. For example, they thought maggots appeared from rotting meat, frogs came from mud, and mice were created from grains and dirty clothes. This idea was supported by ancient philosophers like Aristotle and later by scientists such as Van Helmont and John Needham. However, later experiments proved that this was not true. The biogenesis theory replaced this belief and showed that life can only come from pre-existing life. Francesco Redi, in 1668, showed that maggots came from flies, not from meat. He used 3 jars, each containing a piece of meat, and treated them differently to observe what would happen. In the open jar, flies could enter freely, lay their eggs on the meat, and after a few days, maggots appeared. In the cork-sealed jar, the jar was tightly closed so that no flies or air could enter, and as a result, no maggots were found. In the gauze-covered jar, the jar was covered with a thin cloth that allowed air to pass but prevented flies from touching the meat. The flies laid their eggs on the gauze, and maggots appeared only on top of the cloth, not on the meat. This experiment showed that maggots come from fly eggs and not from the meat itself, disproving the idea of spontaneous generation. Lazzaro Spallanzani further supported this idea by boiling broth and sealing it, preventing the growth of microorganisms. He boiled nutrient broth in several flasks to kill any existing microorganisms. He then sealed some flasks tightly and left others open to the air. After a few days, he observed that the open flasks became cloudy due to the growth of microorganisms, while the sealed flasks remained clear. This showed that microorganisms did not appear spontaneously but came from the air. His experiment provided strong evidence for the biogenesis theory, proving that life arises only from pre-existing life and not from non-living materials. Finally, in 1861, Louis Pasteur's famous swan-neck flask experiment gave the final proof. He demonstrated that microorganisms in the air caused contamination, and when dust and germs were prevented from reaching the broth, no life appeared. Pasteur boiled nutrient broth in special flasks that had long curved swan-shaped necks. This design allowed air to enter but trapped dust particles and microorganisms in the bend of the neck. As a result, even though air could reach the broth, no microbial growth occurred, and the broth remained clear for a long time. However, when the flask was tilted so that the broth came into contact with the trapped dust, microorganisms began to grow, making the broth cloudy. Thus, the biogenesis theory proved that all living organisms arise only from other living organisms, not from non-living materials, completely disproving the idea of spontaneous generation. The germ theory of fermentation was proposed by Louis Pasteur, a French scientist, in the mid-19th century. Before his work, people believed that fermentation, the process by which sugar turns into alcohol, happened naturally or by some vital force. Pasteur proved that microorganisms are responsible for fermentation. He showed that when yeast was present, sugar was converted into alcohol and carbon dioxide, producing wine and beer. However, when unwanted bacteria entered the mixture, they spoiled it by turning alcohol into acids. Pasteur also demonstrated that heating the liquid could kill these harmful microbes, a process later called pasteurization. His experiments established that specific microbes cause specific chemical changes, forming the basis of the germ theory, which later led to the understanding that microbes can also cause diseases. Thus, Pasteur's work on fermentation not only revolutionized the food and beverage industries, but also laid the foundation for modern microbiology and medicine.
The germ theory of disease states that many diseases are caused by tiny living organisms called microorganisms or germs, such as bacteria, viruses, fungi, and protozoa, which invade the body and disrupt its normal functions. Before this theory was developed, people believed that diseases were caused by bad air, miasma, imbalances of body fluids, or supernatural forces. The work of several scientists helped establish the germ theory. Louis Pasteur demonstrated that microorganisms cause fermentation and spoilage, disproving the idea of spontaneous generation and showing that microbes are responsible for many diseases. Robert Koch, identified specific bacteria that cause diseases and formulated Koch's postulates, a set of criteria to prove that a particular microorganism causes a specific disease. Joseph Lister applied this knowledge to surgery by introducing antiseptic techniques, and Ignaz Semmelweis showed that hand washing could prevent the spread of infections during childbirth. The germ theory revolutionized medicine by leading to the development of vaccines, antibiotics, and hygienic practices, transforming healthcare and public health worldwide. Koch postulates are 4 scientific principles developed by Robert Koch to show that a specific microorganism causes a specific disease. These postulates became the foundation of medical microbiology and helped prove the germ theory of disease. First, Koch stated that the microorganism must be found in all individuals suffering from the disease, but not in healthy ones, meaning the germ is always linked to the illness. Second, the microorganism must be isolated from the diseased host and grown in pure culture, so scientists can study it separately. Third, when this pure culture is introduced into a healthy, susceptible host, it should cause the same disease as observed in the original host. Finally, the microorganism must be re-isolated from the newly infected host and shown to be identical to the original microbe. These steps provided clear scientific evidence that germs are responsible for causing diseases, not bad air or other myths. Koch's work was revolutionary. It helped identify the bacteria that cause anthrax, tuberculosis, and cholera, and it laid the groundwork for modern infection control, diagnosis, and vaccine development.